When most hear the words chemical weapons, they think of a strange colored gas that poisons all who inhale it. As everyone knows, chemical weapons are dangerous. But what makes chemical weapons so devastating to the human body at a closer look? At the same time, what lesson can be learned from this advanced understanding? Chemical weapons are defined as a chemical used to cause intentional death or harm through its toxic properties. Most often, they are in gaseous form, but have been known to come in aerosols and liquids as well. Now, with that set up, let's get into the four types of chemical weapons. First up is choking agents. Choking agents are delivered through gaseous form, attack the respiratory system, and have delayed symptoms. Some examples of choking agents include chlorine gas and phosgene gas. Choking agents are inhaled into the alveoli of humans, where they react within the alveoli and bronchi to create hydrochloric acid and hypochlorous acid with water. These acids begin damaging alveoli and blood vessel tissue by damaging the cell's proteins. The proteins that form the body cells are formed through a complex series of forces that hold them together, with long polypeptide chains having many functional groups that all interact with one another. Nonpolar functional groups cluster together away from polar water, between maybe two methyl groups. Hydrogen bonding occurs between the several polar functional groups that contain a hydrogen and an oxygen, nitrogen, or fluorine, like a hydroxyl and carbonyl group. Ionic bonds occur between negatively and positively charged side chains, like an ammonium and negatively charged carboxyl group, otherwise known as a carboxylate group. Lastly, van der Waals forces help fine-tune the forces at play to allow the protein's complex structure to be stable. Proteins are complex but vital to the function of the body. However, the pH change caused by the hydrochloric acid and hypochlorous acid damages the proteins. These acids dissociate in water, and the positive and negative ions pull polar side chains of the proteins apart. They denature the protein, making it useless and unable to perform its job. The denatured protein causes the cell to fall apart, as proteins denature and are unable to maintain their role and shape in the cell. The body tries to respond with blood vessels opening and inflammation rising to quell the damage, but it ends up only filling the lungs with excess fluid, causing further breathing issues similar to what's experienced in COVID-19. At the same time, hydrochloric, hypochlorous, and chlorine gas damage neutrophils and mitochondria of cells to create superoxide, hydrogen peroxide, and hydroxy radicals. These are reactive oxygen species, or ROS for short. These ROS react further with amino acids in DNA itself by oxidizing them, making them lose electrons, and changing their oxidation state. The oxidation state is the number of electrons lost or gained by that element in a chemical combination if the bonds were purely ionic. Essentially, how many electrons are gained or lost by that atom when bonded with another, in a hypothetical. Copper, for example, has an oxidation state of +2 and +1, because when it bonds with a nonmetal like oxygen, it loses its valence electrons and increases its oxidation state. When it separates with oxygen, its oxidation state goes down because it has gained its electrons back and reduced to a more normal state. In a more complicated example, by changing the oxidation state of the atoms in DNA and proteins, the ROS damages the genetic information held by the DNA. The consequences are an increased likelihood of apoptosis and cancer further down the line, as the DNA's amino acids change and the genetic information is permanently lost. Next, let's take a look at blister agents. Blister agents are often delivered as gases as well. They attack the eyes, lungs, and throat. Some popular examples of blister agents from history are sulfur and nitrogen mustard gas. They perform an intramolecular cyclization reaction, where a long chain becomes a ring, thus the prefix cyclo. Then a chlorine ion is created, while the molecule forms an ethylene sulfonium ring. These produce electrophile, electron deficient, sulfonium and ammonium ions. The ethylene sulfonium ion alkylates sulfhydryl and amino groups on DNA, RNA, and protein structures. The sulfonium alkylates by forming a carbonium group to react with DNA, RNA, and proteins with the cyclo portion of their chain. These ions alkylate with specific groups like guanine N7 and adenine N1 and N3. Alkylation involves the transfer or addition of one alkyl group to a new parent chain through either an addition or substitution reaction. An alkyl group is an alkane molecule missing one bond, so it can form an alkyl group with a parent chain. Alkylation can be an addition reaction where double and triple bonds are broken and a new alkyl group bonds to the parent chain. An example is deoxyguanosine in the image undergoing alkylation. Deoxyguanosine is a variation of guanine and a building block of DNA. It also can be done through substitution where atoms and molecules swap, but in this example, it is an addition reaction. The DNA bases, usually the rings of nitrogen or exocyclic oxygens, alkylate first. These cause breaks, called lesions, in the DNA, causing the DNA to be more likely to mutate and become cancerous in the future. The same occurs to proteins, causing them to fall apart and cells to die, leading to further tissue death. Third is blood agents. Blood agents interrupt cellular respiration in the human body's cells. Cyanide ions are created rapidly by blood agents, most famously hydrogen cyanide and cyanogen chloride, after entering the lungs and bloodstream. The slightly alkaline bloodstream breaks HCN molecules into hydrogen ions and cyanide ions. The cyanide ions bind to hemoglobin in the bloodstream. The negative cyanide ion bonds to the positive iron 2 atom in the hemoglobin, the same hemoglobin found in blood that carries oxygen to the body cells. Instead, it creates cyanohemoglobin. The cyanohemoglobin brings cyanide into the mitochondria on the electron transport chain in cells across the body. It prevents the reduction reactions between oxygen and hydrogen at the end of oxidative phosphorylation from occurring in the electron transport chain. It does this by binding to an enzyme called cytochrome C oxidase instead of oxygen. It binds to the copper and iron 3 atoms of cytochrome C oxidase that attract the electrons at the final stage of the electron transport chain. Instead of forming water, it forms hydrogen cyanide and stops cellular respiration. It prevents the 26 to 34 ATP from being made during oxidative phosphorylation and causes rapid cell death as it runs out of energy. Our fourth and final chemical weapon of the day is nerve agents. Nerve agents block impulses between nerve cells and across synapses. They generally cause seizures, muscle paralysis, and loss of bodily control before death. Acetylcholinesterase, or ACHE, is an enzyme used by neural receptors. ACHE manages the amount of acetylcholine, or ACH, a type of neurochemical messenger. It prevents excess ACH and protects its neural receptors from being overloaded. It does so by breaking it down through a reaction called hydrolysis into acetic acid and choline. The carboxylic ester bond is broken and replaced with a hydroxyl group from the enzyme and a hydrogen atom. In summary, adding one water molecule. Hydrolysis is a reaction that involves a larger molecule being broken down into two smaller ones with the addition of water, which is what happens in the breakdown of ACH. The nerve agents are formed once two non-toxic chemicals combine. For example, isopropyl aminomethyl ethyl phosphorite and elemental sulfur react to form a nerve agent called VX. They form organophosphorus compounds similar to pesticides that are released by nerve agents after absorption. These compounds are nonpolar, so they dissolve and accumulate in oily adipose tissue in the body, where they go to interrupt nerve function. The organophosphorus compounds bind to ACHE and prevent them from controlling ACH levels. The excess amounts overstimulates neural receptors for muscles and causes sludge symptoms and death. Now, many of you might be wondering, why is it important for scientists and regular people to understand the chemistry of chemical weapons? The answer is because it is important to learn from history. Chemical weapons use are at an all-time historic low and see little chance of making any major resurgence except for the chance of a large-scale war. Humanity is lucky in this aspect. The knowledge of the chemistry of how these weapons work is simply meant to remind people why these weapons should stay at this historic low. At the same time, it is crucial for medical professionals to be knowledgeable of how to treat those affected by chemical weapons. Anyways, I hope you enjoyed the video and found the information interesting. A Quizlet is in the description of the video if you want to test yourself with the chemistry information from it. Be good people, y'all. See ya.